Okay, hi everyone. Thank you for coming out to my talk. My name is Ruth Rivkin, and hopefully it's not too obvious. I just dumped coffee all over myself. I work with the University of Manitoba, San Diego Zoo Wildlife Alliance, Polar Bears International, and Environment and Climate Change Canada to try to understand how polar bears can survive climate change. And today I'm going to be sharing some of my research looking at the risk of climate maladaptation for Canadian polar bears. First, I'd like to start with some basic polar bear biology and some information about how climate change is affecting the Arctic. So there's around 26,000 polar bears worldwide, and they live exclusively in the Arctic. They are very well adapted to the Arctic environment, but due to recent declines in sea ice coverage and that have resulted from climate change, they are listed as vulnerable by the IUCN. This map here is showing you the polar bear subpopulations, which are management designated units, and Canada has 13 of these subpopulations. That's around 2/3 of all polar bears in the world. And what this means is that Canada has a large role to play in the conservation and management of polar bears. As I said, polar bears are very well adapted to life in a cold Arctic. They have all kinds of physical and genetic adaptations that help them survive very cold temperatures. They rely on sea ice for a lot of their basic biological needs. They hunt on the sea ice for their primary prey sources, which are typically ringed and bearded seals. They use sea ice as a platform for dispersal, and while they can swim very long distances, this is very energetically intensive. And so to get around the landscape, polar bears prefer to walk and move around the ice. There's other things that happen on sea ice. Mating happens exclusively on sea ice, and until recently, denning for females happened on the ice. Although recently, due to declines on the sea ice, polar bear females are denning on land more and more often. I'm sure everyone here is familiar with climate change and believes that climate change is happening. But what I want to share with you is that climate change is happening at an extremely rapid pace, 4 times the pace of the rest of the world in the Arctic. And to illustrate this, I'm showing you a satellite image of the North Pole in 1979. And I've just highlighted the ice edges in red here. And I'm going to show you the same image again in 2022. And what you can see is that the Arctic has lost approximately 2/3 of all ice cover in the summer. And this is enormously problematic for species that depend on sea ice for survival. We know that sea ice loss has been associated with a bunch of different ecological changes in polar bears. Polar bears population abundances are declining, and this has been directly correlated to sea ice loss. As sea ice is declining, polar bears have to spend more and more time on land, which means that they are fasting. While they're on land, they don't eat typically, and if they do eat, they can't get enough food to sustain their energetic needs. And so while they're on land, polar bears will lose up to 1 kilogram of mass per day until they go back on the ice. And in some regions of the Arctic, especially in the southern areas, this fasting time can be anywhere between 4 and 6 months. And as climate change continues to warm the Arctic, we know that at some point we will reach a limit where polar bears can no longer fast for this amount of time. And so this raises a really important question of, can polar bears survive climate change? Polar bears have 3 options in the face of climate change. They can disperse to habitats that may be better suited to their lifestyle, but this is challenging because they're already as far north as they can realistically go, and so there's limited areas for them to disperse to in order to survive. They can potentially adapt to climate change, so change their genetic structures and become better suited to life in a warmer Arctic. And they could go extinct, or at least locally extirpated in regions of the Arctic where they can't survive. And so we think a combination of these 3 things will happen for polar bears. But my research explores the adaptive potential of polar bears under climate change. And to do this, we designed an 8K SNP chip for polar bears. This is my co-author, Josh Miller, holding an example of that chip. And we had 3 primary goals for this chip. Our first was to assess standing genetic variation across the subpopulations in Canada. And this allows us to assess the capacity that subpopulations might have for adaptation. because subpopulations with more genetic variation have more raw material to be able to adapt to changing environments. We also wanted to examine genetic forecastings under climate change scenarios to get a sense of which subpopulations might be better suited genetically to climate change environments and which populations might be more vulnerable to climate change. And lastly, we wanted to assess gene flow with grizzly bears. We know that hybridization is possible between the species in the wild, and gene flow can potentially introduce warm adapted alleles into the polar bear population that might help them survive climate change. But too much gene flow might swamp out all that unique genetic variation that makes polar bears polar bears. And so we wanted to explore how often gene flow happens between the species. So first we assessed genetic variation across the Canadian Arctic, and we did this in 411 polar bears, sampled from 13 subpopulations, and I'm showing you a map of those subpopulations. The pie charts represent genetic structure across the Arctic, and this gives us an indication of how polar bears are related to one another. We found 5 clusters, which is consistent with what other studies have found, but what we identified was a unique genetic cluster way up in the high Arctic in Norwegian Bay, and so this has not previously been confirmed, and so this suggests that the bears in this area have some unique genetic variation going on. We then assessed genetic diversity to get a sense of adaptive capacity. And what you'll notice is that here I'm showing you observed heterozygosity, but this was true across all measures of diversity we looked at. Norwegian Bay has a lower amount of genetic diversity than our other subpopulations. And we know that polar bears already have low genetic diversity compared to grizzly bears. And so this result suggests that Norwegian Bay might be especially vulnerable to climate change through reduced adaptive capacity. And it's interesting that when we looked at private alleles to get a sense of which subpopulations might contain genetic variants that are unique to those specific subpopulations, Norwegian Bay also had the most number of private alleles. And so this suggests that not only is this subpopulation genetically unique, but it also might be particularly vulnerable to climate change. We next assessed the genetic mismatch to climate change, and we did this by using a machine learning method that allows us to calculate genetic offsets. And so to translate that into something that's more understandable, we took the genetic data that we collected, and we mapped the allele frequencies out under different environmental scenarios of temperature and ice condition. We then paired these with climate models showing different levels of warming under different emission scenarios, from low emission and lower warming to high emission and higher degrees of warming. And we then predicted the allele frequency change under those new environments. And here what we're assuming is that polar bears are currently adapted to their environmental conditions. And so any shift away from this optima is presumed to be maladapted. And so larger genetic shifts suggest a larger genetic mismatch in those polar bears. We then summarize these across subpopulations to get an indication of vulnerability to climate change scenarios. And so using the same polar bear set that we looked at for genetic diversity, we mapped out the genetic offset or genetic mismatch across the Arctic. And so here what I'm showing you is a map of our study area with our polar bears in black. And the areas of the Arctic Ocean that are covered by sea ice usually have been color-coded based on the risk of maladaptation, with blue being a lower risk and red and yellow being higher risk. And what you should notice is that it's in the high Arctic that we see those genetic mismatches. And this is the same region where we also saw lower genetic diversity and an increased genetic structure signal. We also found that genetic offset was associated with subpopulation abundance. And in particular, these 3 subpopulations with the smallest abundances are the ones found up in the high Arctic. And so what this suggests to us is that the risk of maladaptation is highest in the small, isolated populations in the high Arctic. And so this might be an area of the Arctic that needs additional conservation efforts aimed at preserving that unique but vulnerable genetic diversity. Lastly, I'd like to share some findings on polar bear-grizzly bear hybridization. As I mentioned earlier, gene flow between the species can potentially benefit polar bears by introducing warm-adapted alleles into their population, but it could more likely swamp out that unique genetic variation that makes polar bears their own species. And so we wanted to get a sense of the rate of hybridization. We know that hybridization is possible. There have been 8 documented cases in the Western Arctic. And these all originated from this family. So we had a polar bear female who mated with 2 grizzly bear males. She produced 4 hybrid offspring. These are F1 generation hybrids, 50% polar bears, 50% grizzly bears. And one of her daughters then mated with those same 2 grizzly bears, including her father, to produce a back-crossed grizzly bear hybrid generation. And these would be 75% grizzly bear, 25% polar bear. And so using these hybrids that we knew about, we validated our SNP chip and looked to see how often hybridization might be happening but might not be accounted for. And that's because it can be really hard to tell a hybrid from a dirty polar bear. And so we might miss hybrids when we're doing visual scans. So we scanned 800 bears, and what I'm showing you here is a PCA of genetic relationships. And the biggest thing to take away from this is that bears that are genetically similar cluster together, and we have a really good split between our species. So we have grizzly bears in green and blue, and polar bears in orange. And what you can see is that there's no overlap between species, so we can detect separate species. When we plot our hybrids in here, we see exactly what you would expect. Our F1 hybrid generation is in the middle, exactly between the species, and our back-cross generation is a little bit closer to the grizzly bear side of things. But what I want you to take away from this is that we didn't find any new hybrids. This is our data set. And so to us, this suggests that hybridization remains quite rare in the Arctic, at least in the time frame and in the area that we looked at. So right now, we think that hybridization is probably not a source of adaptive genes, but it also does not risk the loss of polar bears as a species. But what's important is we now have this relatively inexpensive and easy to use resource that we can continue to monitor the polar bear population so that we can see if hybridization begins to happen more often. And so in conclusion, the conservation of polar bears will rely, at least in part, in preserving the adaptive potential of the different subpopulations and minimizing those risks of maladaptation. But ultimately, the biggest thing that we can do to benefit polar bears right now is reduce our emissions and try to mitigate as much future warming in the Arctic as we can. And so I have a ton of collaborators, like too many to throw up on the slide, but I am happy to take any questions or comments as well. Thank you. Yeah.
We make a strong argument that the high Arctic populations are particularly important for conservation. We talked about additional management. And I suppose I'm just curious, not knowing anything about polar bears, is there something obvious that can be done in terms of managing those high Arctic populations?
Yeah, so one thing that's happening more, or just being discussed more in the Arctic, is shipping through the Northwest Passage, and those bears in the high Arctic are primarily found in the shipping areas. So making sure that if shipping does begin to happen through the Arctic as the areas of the Arctic become ice-free, that we can preserve as much of the habitat of polar bears is one way that we could potentially target those polar bears. Yeah, so we did look at that. I didn't share that here. So we did some assessments of continuous and spatial population structure. So we see essentially that there is the presence of isolation by distance, where bears in southern regions of the Arctic are not necessarily sharing genes with bears at the high Arctic. And so while gene flow does happen, because polar bears can move millions of kilometers, like a single female has been documented to have a range size of over 1 million square kilometers, but they are limited to some degree by distance and also by geological barriers to their dispersal patterns. And so we don't think that gene flow between the high and low Arctic is possible right now, but things may change as the ice continues to decrease.
Good. Okay. Hi, everyone. My name is Kat Sullivan. I'm a PhD candidate at Marquette University, and I work with the Milwaukee Public Museum for my dissertation research in integrating community science, field surveys, and natural history collections in pollinator research for conservation. So, as we know, there's been a lot of recent studies that point out the dramatic declines of insects broadly and at a rapid pace, and many of these are attributed to human-mediated developments. So from land use intensification to more intense climate change events, insects are extremely vulnerable to many of these symptoms of climate change. But insects are also extremely diverse, and how these declines are distributed across all the different groups of insects is extremely variable, and we don't have a lot of information about how this will vary across space and time as of yet. And so I do my work in Wisconsin, which is also a very ecologically diverse state in the Great Lakes. And we are primarily focused on pollinator insects and trying to track how their ranges are changing over time. And so in the state of Wisconsin, there are 4 federally endangered or threatened butterfly species, many of which have shown dramatic decreases in their range or shifts due to climate change. And butterflies often are highly tied to their host plants, so the threats also affect specific native plants as well for the caterpillars, which needs to be taken into consideration in future conservation plans. So my main research questions are, how does pollinator plant availability impact butterfly diversity across Wisconsin, and how has that changed over time? And to do that, we need a lot of biodiversity data that's going to cover a wide range of space and time. But unfortunately for insects, because they're also so diverse, they are the most data deficient group compared to other larger groups that have less diversity, like vertebrates, vascular plants. And so there's an extreme data deficiency in terms of records that can be used to address these questions for insects. So I also am aiming to find new ways to integrate biodiversity data sets in modeling and trying to assess these questions. So a great way to do this is by engaging the community and deploying community scientists out into participatory science projects for research. And so at the Milwaukee Public Museum, we've established a Wisconsin statewide community science network, partnering with many institutions, nonprofits, science centers, universities across the state, all with the same goal of expanding community science across the state and educating the public about biodiversity and some of these concerns with conservation. And so we've focused primarily up till now on butterflies, and we have contributed many new materials like identification guides to help make these surveys easier for people who are not experts in the field, and also leading guided walks across the state. So there's many ways to contribute to scientific research. One that many people are probably familiar with is iNaturalist. And so we have an iNaturalist project now, has close to 25,000 observations, where anyone is able to contribute any biodiversity observations that they have, and it can be used into our data set and also for other researchers interested in similar questions. Another method that we have started to develop is the Pollard Walk protocol, which is a standardized survey method that uses observation-based protocols. And so this was developed in the UK in the 1970s by Pollard. The walk protocol is very simple. You choose a route that has ideally a variety of habitat types that you can access multiple times over the year after year, to repeatedly survey. It is just a visual-based survey, so no collecting is required. And we have since established sites in over a dozen areas across the state. So I'm just going to briefly go over my methodological framework here for integrating these different types of unstructured and structured data sources in trying to model how butterfly occurrences are varying over the state. So first, I needed to aggregate the plant records as a predictor for whether or not we see different butterfly species across the state. And so we have three main data sources that we're pulling from. So the first, as I already mentioned, were the observation data sets. So these are mainly digital records, checklists, and that is what the iNaturalist data set is consisted of entirely. Then there's also specimen records from the museum. Our museum in Milwaukee has the largest physical specimen collection for the state of Wisconsin. And we also can use other repositories like Symbiota Scan to add in additional records from other collections. Then we also have our systematic surveys through Pollard-based, which is our standardized survey method. And then GBIF, which is the Global Biodiversity Information Facility, has all of these types of records as well. So pooling all of these together, we developed a data set for all of the plants in Wisconsin and then all of the butterflies. So next step in building our model, we use Maxent in R for all of the plant families using environmental predictors of mean annual temperature and mean annual precipitation across the state. And then we use those plant family presences, aggregated them all together to get a proxy for richness across the state. And then we repeated that same process again with the butterflies, but this time using plant richness as a predictor for butterfly species occurrences as well. So then we repeated stacking here with the butterflies, and then we wanted to look at how the aggregated richness of the plants to the butterflies was going to correlate. And so we expected that because butterflies are often tied to specific plants, that an increase in plant diversity would result in an increase in butterfly diversity, and they'd be highly correlated. So as this work is still in progress, I'm just going to point out a few interesting things that we found in a case study here with the grass skippers or the Hesperiidae. And so first I'm just going to talk about those observation and occurrence data sets of the visual surveys through iNaturalist, and then also the specimen vouchers in museums. So this is a histogram showing the spread of the data here for all of the records across Wisconsin. And there's very clear differences between these two data sets. So our preserved specimens there in blue from the museums have a much wider temporal scale. We have records dating back to the late 1800s with really a peak in occurrences in the 1960s, 1970s, then a pretty drastic decline in the 21st century. But this was supplemented by the increase in community science and observation data through the platform iNaturalist, eButterfly, other projects that aimed to survey butterflies. And so then we end up with this enormous spike in records that have been available over the last 10 years. And so we had over 5,000 unique records for 76 taxa. That was split pretty evenly between the preserved specimens and the human observations. All right, so using this Hesperiidae case study, our plant family richness, we had 44 plant families. This is a species distribution model where the darker green corresponds to higher predicted plant richness. And so as the plants we used the environmental predictors for in the south where there is higher overall mean temperature, we have higher richness and then a pretty stringent gradient going north. On the right there is just an overlay of all the sampling locations for the plant families to show that we did have a pretty even and complete coverage of the state through these data sets. But there are obviously some places that are more heavily sampled, which we used different grid sampling methods to try to account for. And so using a stack of all the plants, then we were able to use plant richness as a predictor of the grass skippers across the state for each species, and then aggregate those as well. But I found a problem in trying to aggregate both of these data sets in that the occurrence and the observations were giving pretty different results. So on the left there is the occurrence-only data set. Sorry, the text is a little small, from the museum specimens. And so again, richness that's darker green is higher, and then we have the overlay of the points for sampling. So definitely a little bit spotty in sampling with the museum specimens, and again, this spans a wide range of time, but we're just seeing a couple of hot spots of plant richness, and plant diversity was the #1 predictor, with accounting for about 65% of importance for this data set. Then contrast that on the right with our observation data set, where we have a similar distribution of sampling, again, usually highly concentrated around major population centers where people are, but we have a different hotspot there for richness in kind of the central part of the state. But again, diversity was also the highest predictor in terms of variable importance, but just a little bit lower at 45%. So then when I went to do the correlation between the butterfly richness and the plant richness, as you can kind of tell from the previous map, we got pretty different results. So on the left is the occurrence record data set, correlating plant richness on the x-axis and butterfly richness on the y-axis. We had basically no correlation found in this data set of occurrence records. So there were some areas where there was really high plant richness but pretty low butterfly richness, and then the inverse was also true. But then in the observation record data set, which is a much less constrained temporal data set, we did find a pretty weak correlation, but much different than the occurrence record data set. So there is a little bit of signal coming out if you're just looking at an observation data set, which is pretty interesting. And then if you were to combine both data sets, there's really no signal at all, and it's just all noise. So it's really hard as of right now to integrate these two data sets. The only consistent thing is that plant diversity is clearly a very important predictor. And so I looked at all of the individual plant families as well, and I think data is just a little bit too limited to really draw any conclusions at this stage about which plants are the most important ones, but that's something that I'll be looking for in the future. And so going back to those surveys that I mentioned, how are we able to integrate these standardized surveys with these unstructured data sources if there's already so much discord between those two data sets as it is? So going back to the full butterfly data set, we looked at just which species were present, and not even full coverage is found in all of those data sets. So out of 124 total taxa found, we had 40 common species that were found in all 3 data sets, but each one also had unique species still. And between the specimen data set and the naturalist data set especially, there were a lot of unique species that were only found in those data sets. But the surveys seem to be promising for maybe integrating these two better, and we can use these models to predict areas where we can go and concentrate more effort, especially in our increasing community science program. So the Wisconsin Butterfly Monitoring Network, which I mentioned, was only launched in 2019, a little bit of a gap with COVID, but now we're increasing with sites and data each year. So looking at where our sites are on the left with the blue points, we see that those aren't necessarily the same points where we're finding that high hotspots of richness. So this is pointing out ways for us to try to go to these sites, find more partners, and collect more data to try to get a more robust data set. So with that, I would like to thank all of my collaborators, the museum, and the community across the state. And if you're interested in any more of our work, I'm happy to chat later or take any questions. Thank you. Yeah, so the question was, were there any sites that there is overlap? So I haven't looked specifically at that, but I do know that a lot of these sites have been kind of historically surveyed more than others, and so we have seen some sites where there is complete congruence, but yeah, looking at kind of as a total picture, it's really hard to find places that have those long-term data sets where there's been a pretty long historical record of surveys and then newer records of observations, and then having to take into consideration all of the environmental and land change. So yeah, that's definitely something we're going to look to try to figure out too. Okay, thank you.
Well, thanks everyone for sticking around for another Skipper Talk. I'm really excited to tell you today about some of the work I've been thinking about as part of a Fish and Wildlife Service grant that I have. My name's Josh. I'm a postdoc at the University of Wyoming, and I'm a part of the Modelscape collaboration with funding from NSF EBSCOR. And I think all of us are becoming more and more aware and having a little bit more and more despair all the time because of all the threats that wild populations are currently facing in the world, right? It's getting to be the point where nearly every wild population is facing at least one set of threats, and oftentimes many threats. And this is really, really concerning because we value biodiversity. I don't know, many of us are here because our passion for science was initially started by loving nature or skippers or polar bears or anything like that, right? And so I'm really passionate about trying to use what I've learned in evolutionary biology and in genetics to help managers better conserve wild populations. And at least in the United States, there have been a few success stories through kind of federal actions, right? So the Endangered Species Act just went through its 50th anniversary. I'm not going to say that it's been this magic cure that's been successful in all cases. Many endangered species have gone extinct, and that's terrible. But there have been a few really bright beacons of hope, suggesting that this program might allow us to first, recover populations that are imperiled, but also by acting progressively to prevent organisms from being listed even in the first place. Now, a key component of the Endangered Species Act is that in order to be a part of the program, an organism or a lineage needs to be identifiable lineage. So whether that be a species or subspecies or a distinct population segment, it has to be known to be its own evolutionary lineage. And for many organisms, this isn't an issue, right? We've have a long history of doing taxonomy and using genetics to inform taxonomy throughout North America. But there are a few geographic areas and some lineages where this becomes more difficult than others. And one geographic area I want to focus on today is the Great Basin. The Great Basin is a large region in the western United States that's found between the Sierra Nevadas and the Rocky Mountains. Throughout this region, there stretches a series of mountain ranges separated by low basins over and over again, dozens of mountain ranges throughout the area, and they all run north and south. And the key thing I want you to take from this is that within a very close geographic space, you can have dramatically different environmental conditions. And this potential for local adaptation sets the scale or sets the stage for the possibility of diversification of very rare, localized, and patchily distributed forms. And one organism that kind of fits the bill for this is the sandhill skipper, which I'll be talking about for the rest of the day. This is one of the taxonomically most diverse, putatively diverse butterfly species in the United States. Within the state of Nevada alone, there are 9 different subspecies that are found based on morphological taxonomy. And so this is a paper by Austin from 1987 where he mapped out the different subspecies across the state. And I've kind of colored or labeled or put the symbols of all the different butterflies on the left-hand side there. And so you can see that there's definitely some morphological variation in the wing coloration or wing patterning, perhaps, right? And that was the basis of this. This is all, this photo here is all the dorsal view from females. And I'm going to point out one population in particular and one subspecies in particular, and that's the one in the bottom row in the center, and that is the bleached sandhill skipper. It is known from only one population in the northern part of the state, right on the Oregon border, and it has a very unique morphology compared to the rest of the different subspecies. And so it's been kind of the focus of thinking about conservation efforts in this group. To give you a little bit more background, this is the spot in northern Nevada where it is. And so in this little valley here, there is a lot of geothermal activity, and so hot springs coming out of the ground, which the plant communities are dependent on. There's two main plants there, a salt grass that the caterpillars are dependent on for consumption, and then lots of rabbit brush, which the adults are dependent on for nectar sources. And the reason why folks are so concerned about this population is because recently the Bureau of Land Management permitted a geothermal facility just on the other side of the highway. And folks are worried that by putting in this geothermal facility, it will disrupt the water flow on the other side of the highway, disrupt the plant communities, and lead to the extirpation of the butterfly. And so a few years ago, the bleached sandhill skipper was petitioned for being listed on the Endangered Species Act because of this. However, this is complicated by some genetic work that I did back in 2015 and published. And so I'm going to show you just a quick rundown here and focusing in on that one population. This is the result from mitochondrial DNA, CO2 sequences, and you'll see that there's a single haplotype at that Pueblo Sioux location, the K haplotype, but it's not private to that population. It's also found in the population in Oregon and Northern California. And then when looking at a structure plot based on AFLPs, again highlighting that Pueblo Sioux population, it's kind of a mess, right? And so we can't really say a whole lot on this plot here. Each bar, vertical bar, is an individual, and they're color-coded by which ancestry they have. And I think I take away two kinds of main take-homes from this slide here. One is that it's really scary to look at figures you made over a decade ago. And the second thing is that we can't really say anything about the taxonomic uniqueness or identity of the bleached sandhill skipper. And I don't know whether or not that's because it's not its own evolutionary lineage or because I just didn't have the power back then to identify that. Adding on top of that, since that last work, there have been additional populations within close geographic proximity to that focal Pueblo Sioux or PS population. And we just aren't sure exactly what that identity is. Some folks have said that they might be hybrids. We're just not sure. And so what we wanted to do was to investigate this a little bit further using more modern techniques. And so I worked very closely with Laura Enders at the United States Fish and Wildlife Service, Reno office, to develop the entire research plan for this project, identify the key gaps that were missing for their listing decision that will be coming up. And we had three main kinds of questions. One was, what's the genetic ancestry of those unknown populations? This is really relevant. Do we have one population to work with for conservation, or do we have three maybe? The second is the bleached sandhill skipper a distinct evolutionary unit? This is critical for understanding whether or not this should be listed. And then as kind of a proxy for risk, albeit problematic, how does this genetic diversity of that focal population compare to other populations? And so to answer these questions, I generated genotyping by sequencing data that's analogous to double digest RADseq data. And what I will show you on the next few slides is the summary of variation across about 20,000 genetic markers. The first question was about those unknown populations. And here I'm showing you a principal component analysis of that variation for those 4 geographically proximate locations. Here we have the first and second principal components on the axes. Each individual point represents an individual butterfly. And what you can see here is that the first principal component separates the Alta Canyon other subspecies, the alkaliensis subspecies, away from the 3 other populations very strongly. And in fact, there's 14.7% of the variation explained on that first principal component is actually fairly large. And then the 3 other populations kind of separate out to a lesser extent on that second axis. And this tells me that the 2 unknown populations strongly ally with bleached sandtail skipper. This is really great news because now we know that there's 3 populations that we can work with for conservation. And we had no idea about this going into it. Taking a broader taxonomic view, we also sampled many of the same populations from my original paper. And here on the map, you can kind of show the distribution of ancestry that was sampled across the different lineages and kind of ecoregions. And this is a neighbor joining tree based on pairwise FST. Each point on the tree represents one of those populations on the left. And the distance between the different tips represents how genetically similar or different they are. And what you can see here is that those 3 populations, Pueblo Slough, and the 2 unknowns, all are on a very long branch, all by themselves, again suggesting that this is a distinct evolutionary lineage. You also might be standing some of the other tips and notice that many of the other subspecies that I mentioned up there don't actually cluster out together. So it's not that the new genetic sequencing methods just solved all the problems taxonomically in the system, but at least for the bleached sandtail skipper, it appears that we have a solid answer. Finally, I'm just going to show you the results of kind of a distribution of the genetic diversity metrics. Here I calculated nucleotide diversity with angst. The Pueblo Slough population had the lowest genetic diversity of all the different populations, but it was kind of within the range of all those other populations, so maybe not quite as concerning as it could be. It's within kind of what you might expect, albeit on the lower end. And so based on all of the results and the unique morphological variation that we see in this system, the Fish and Wildlife Service found in their 90-day listing decision that the bleached sandtail skipper may be warranted for listing. And so that was sometime last year when this 90-day decision was made, and later this fall, they will make a final decision on what will happen. And so I'm really proud of this work because I feel like it directly contributed to whatever their decision is going to be. I have no idea what they'll decide, but at least they have the best information that they need to move forward with this. And just as a final kind of thought, I really want to of these 9 different putative subspecies, which may or may not be real, 7 of them are on the Nevada Natural History Watch List and are currently being thought about for conservation. And so there are many, many populations all around the world that we just don't have any information about. We don't know whether or not we should be conserving them or not. And we as evolutionary biologists should be empowered to help managers and work with managers to solve these questions. And it's only possible if we can work directly with our agency contacts and implement them in every part of the process. And I think we should all strive to do that a little bit more. Thank you.
Hi, everybody. My name is Tony Wilson. I'm from Brooklyn College in New York. And the work that I'm going to be talking about to you about today is a genetic investigation of what is apparently a fairly dramatic population collapse of a coastal population of seahorses in southern Portugal. There are 2 dominant methods that are used to assess population structure and distribution in the context of conservation. The first of these is simple visual census surveys. Often these are done with a standardized methodology. As example here, elephants in the Serengeti from an aerial survey. And essentially, using the same type of methods over time allows conservation managers to estimate the population sizes and changes in the distribution of species over time. This basic method can also be applied in aquatic environments. Researchers use standard fishing trawls, or they can use underwater visual surveys. But one complication in an aquatic environment is the fact that it's a three-dimensional environment where a lot of the areas are actually unsurveyed. So subtle changes in the onshore-offshore migration or depth profiles of species can be inferred as fairly dramatic changes in the population. Now, a method that's become much more common these days is using genetic assessments to estimate the population size and distribution of species. This method is complementary to these basic visual census surveys, and I guess we're all familiar with this. We take samples, either tissue samples or fecal samples from the wild. We do a variety of different molecular techniques, and then we collect summary statistics that allow us to estimate what's going on with the populations in a natural system. As I mentioned, the research system that I'm going to be talking about today is the Ria Formosa in southern Portugal. It's one of the largest coastal lagoons in Europe. 95% of this lagoon is protected. It's a national park in Portugal, and it's also protected by a variety of different national and international treaties. Why is it protected? It's a really important migratory area for migratory birds, and it's also an incredibly important spot as a nursery habitat for a diversity of different aquatic species. Something that somewhat surprises me is that although commercial fishing in the Ria Formosa is not permitted, artisanal fishing and sport fishing is allowed, and there's also quite extensive shellfish aquaculture that's going on in this apparently protected habitat. There's also Faro is a town of about 80,000 people, which is located in the Ria Formosa, which means that there's the potential, at least, for some pretty dramatic anthropogenic impacts. The Ria Formosa is also home to the two common European seahorse species, Hippocampus cucullatus there on the top, and Hippocampus on the bottom. The first extensive spatial survey of these two seahorse species was carried out by Jahnew Curtis in 2001 and 2002. Jahnew carried out a very extensive underwater scuba survey over several years and found that the estimated population size based on her census was that the population size of Hippocampus cucullatus was around 2 million, and Hippocampus was about an order of magnitude smaller at around 200,000 individuals. This research established the Ria Formosa as one of the most rich habitats for seahorses in the world. The densities of seahorses in this area are among the highest ever recorded, and this attracted a lot of interest from conservation managers. Now, unfortunately, only a few years after Jahnew carried out her survey, Europe experienced its highest heat wave in more than 500 years. I was living in Europe at the time, and it was very dramatic. It actually ended up, there's an excess of about 70,000 deaths that occurred. Air temperatures in Europe during this heat wave were about 20 to 30% higher than normal. And perhaps not surprisingly, average sea surface temperatures also were measured at 1 to 4 degrees Celsius higher than normal. This heat wave has been associated with mass mortality in Mediterranean benthic communities. And relevant for my research today, and also quite a pressing issue, is that there have been recurrent heat waves in Europe in 2006, 2010, 2015, and every single year since 2018 in southern Europe. So some pretty dramatic changes. When Ian Caldwell returned to the Ria Formosa in 2008, he carried out a very similar spatial survey and documented that at that point, Hippocampus cucullatus's estimated population size was only 100,000 individuals, which was a decline of about 95% over the previous estimates from Jahnew Curtis. Similar decline was detected in Hippocampus, about a 73% decline. And this decline wasn't restricted to particular locations in the Ria Formosa. Ian actually found that Hippocampus cucullatus, which is the species that I'm going to be talking about today, was entirely absent from 21 of the 32 sites that Jahnew had surveyed only a few years before. So understandably, this attracted a lot of concern both scientifically as well as in the popular media, about this collapse of a really important seahorse habitat in Europe. As I mentioned, I'm going to be focusing on Hippocampus cucullatus, and just to orient ourselves, this is a plot of the census data on the top and the repeated heat waves that have been experienced. So we were fortunate that a group of local researchers at the University of the Algarve have been continually sampling seahorses, both before, during, and after this period of apparent population collapse. So we have 260 samples of Hippocampus cucullatus from across the entire period. We also have 63 outgroup samples from 2 neighboring populations, which didn't experience major population declines during this period. And we carried out some pretty traditional molecular methods. We sequenced the mitochondrial DNA control region and genotype 12 microsatellite loci across the entire sampling period. Most of you, I'm guessing, are probably familiar with haplotype networks. So this is just a general haplotype network. The size of each of the haplotypes indicates the frequency of that haplotype, and the number of steps between each haplotype indicates the number of mutational steps between those particular haplotypes in the population. One thing that's a little bit different here from haplotype networks that you may be familiar with is that this haplotype network represents all of the haplotypes that we detected across the full sampling period. So it's a really good way for us to show how things changed over time. On your right, there's also some summary statistics. Probably the most important here would be allelic richness, A, and haplotype diversity. So what I'm going to do is I'm going to go through what we detected over the period of time. So that's 2000. This is 2004, 2007. So if you look at those data, obviously it went very quickly. If you think that not a lot happened, really not a lot happened over this period. So to summarize, the allelic richness throughout this period, even though there was an apparent population collapse, was sustained. Haplotype diversity was very high throughout this entire period. And when we compare those numbers to neighboring populations, they are comparable or perhaps even a little bit higher. So there is, in contrast to what we detected in the census data, there's no significant temporal change genetically and no significant loss of genetic diversity, which was our primary concern, that a bottleneck like this could actually have long-term consequences. When we analyzed population spatial structure, we also didn't detect any evidence of spatial structure between the Rhea formosa population and the 2 neighboring populations, which is important in terms of the conservation of the species. For the interest of time, I'm not going to be able to go into the microsatellite data, but needless to say, the pattern of genetic diversity at the microsatellites was entirely consistent with the mitochondrial DNA, no significant temporal change, and no significant loss of genetic diversity. And again, no significant spatial structure, which suggests to us that the Rhea formosa population may be part of a regional metapopulation where there's gene exchange occurring among populations, which could potentially give the population additional resilience against these types of climate insults. Now, those are summary statistics. Another way that we can look at data such as this is we can estimate demographic parameters. So we used a software called approximate Bayesian computation to estimate patterns of migration, divergence, and effective population size in this population. What's important is that the effective population size is often orders of magnitude lower than the census population size. So this number is really important in terms of a long-term management. We modeled an ancestral population, 3 separate populations that formed a metapopulation exchanging genes, and a collapse in the Rhea formosa population as we inferred from the census data. Now, when we did this, all we're looking at here is the effective population size. What's very, very important when you look at this is if you compare the ancestral population size of Rhea formosa before the population collapse, the effective population size was estimated at 200,000 individuals. And the more recent period since the population bottleneck inferred from census data, the effective population size has actually increased to about 300,000 individuals. And when you compare those 2 numbers again to our neighboring populations, the effective population size within the Rhea formosa actually exceeds that of the neighboring populations that we're looking at. Again, quite interesting. Now, finally, even though the population change was quite dramatic. We're dealing with a period that's relatively short, and so it's important to ask the question of, is there sufficient statistical power to infer a bottleneck if a bottleneck indeed had occurred? So what we did is we simulated a variety of different scenarios and looked at whether or not we would have sufficient statistical power to infer the population bottleneck. And what we found was, remember that the effective population size in this case is 200,000 or 300,000 individuals. We would have sufficient statistical power to infer a population bottleneck to an effective population size of 500 individuals. So a much smaller population. Clearly, we're nowhere near that. Why is 500 important? Because 500 to 1,000 individuals has been suggested as the minimum necessary to maintain the evolutionary potential of a population. Okay, so to pull our data together, I'm again showing the census data at the top. We have this dramatic change in the population size. However, in contrast to that, there has been no evidence of a degradation of genetic diversity, which would be associated with a decline in the effective population size in this area. Interestingly, these types of surveys have continued over time. And so Miguel Correa has led a variety of spatial surveys in the years since this apparent population collapse. And in 2012, Miguel documented that the size of the hippocampus cucullatus population had increased to around 650,000 individuals based on the census survey, suggesting a rapid recovery, almost too rapid in my opinion, very, very fast. Unfortunately, in a paper that was published just last year, Miguel found that the population size, again, had declined to around 100,000 individuals. And if you look at this together, clearly there's something very dynamic that's happening with seahorse populations in the Ria Formosa. Importantly, the declines in the population size that we're inferring are often associated with these climate events that we're detecting. And as I mentioned earlier in my talk, every year since 2018 has been another heat wave within Europe. And so if there is a correlation between heat waves and declines in population in this particular area, the fact that there's been this sustained period of climate warming over the last 5 or 6 years is a real cause for concern. So we're really hoping to go back out and to continue to survey the seahorses over this period. In addition, I'm also collaborating with Rita Castillo, who's also at the University of Algarve, and we're broadening beyond our microsatellites and mitochondrial DNA to do some SNP typing in this population as well. There's a question of whether or not this is necessary given the levels of genetic diversity that we're observing, but we still have the samples, and so we're going to give that a try just to see what we infer. I would like to acknowledge Alex Nadar from the University of Constance, who was very helpful in developing the approximate Bayesian computation model, as well as Ian Caldwell that helped in interpreting the census data that we saw. And I'd be happy to take any questions you have.
Hi, I'm Aya. I'm a PhD student from Aarhus University in Denmark. I'm very happy to be here today to present some results from my newest paper and provide some genomic insights on delineating appropriate conservation units, this time using Coragonid fishes as a case. So these two, the North Sea Houting and the European lake whitefish, they are salmonid fishes, also called whitefishes. And the North Sea Houting is differentiated from the lake whitefish by its elongated snout and its unique capability of tolerating high salinity oceanic seawater. Both of these are threatened by various anthropogenic activities, and especially the Houting has experienced drastic declines. It was previously found in the entire Wadden Sea system, including tributary rivers in Germany, the Netherlands, and in Denmark. But today, the Houting is found in just a single river called Vidåen in Denmark. Millions of dollars have been spent trying to improve habitat conditions and reintroduce the Houting back into its former range. But at the same time, taxonomic confusion has cast doubt on the actual conservation value of the Houting, and in that way also the necessity of continued investment into its preservation. So essentially it is being questioned if this North Sea Houting is even an independent species, or if it is just like a morph of the much more common lake whitefish, which is found in multiple countries. I say that given the existence of more than 20 species concepts and the fact that species boundaries can be ambiguous at best, delineating conservation and management units solely based on taxonomy can be problematic. So I have actually disregarded the question of taxonomy entirely in this case. And instead I'm adopting this framework from Crandall et al., which aims to categorize population distinctiveness based on concepts of genetic and ecological exchangeability over time. I've tested the hypotheses of this framework using dense rat-seq data in combination with whole genome sequencing data from 6 populations of European lake whitefish and a single extant population of North Sea houting. And going forward, this North Sea houting population will be marked with this tiny fish in order for you to keep track. If we start out by addressing the question of genetic exchangeability, at a recent timescale, this comes down to a simple question of current reproductive isolation. I'm not going to go into much detail. You'll just have to trust me when I say that the North Sea houting currently experiences no gene flow with any lake whitefish populations. The question of genetic exchangeability on a historical timescale is a little bit more tricky. Inference of demographic history using multiple different methods, including multiple sequential Markovian coalescent, or simply MSMC, as shown here. Well, they suggested that all of these analyzed populations belonged to the same ancestral population, which experienced quite a significant expansion sometime around 20,000 years ago. This coincides nicely with previous analysis and with fossil data showing that whitefish recolonized Denmark essentially around the end of the last glaciation. And either prior to or immediately after this event, they went through an expansion. Sometime during this process, population divergence also began. This tree mix topology placed the North Sea houting as the most ancient branch. And it's curious here that it actually appears as if the houting diverged from the other Danish lake whitefish populations even before the German lake whitefish population did. And it's interesting because the German population arc is not just from a different country, it is from a different ocean basin altogether. It is a Baltic Sea population on the east side of the Danish peninsula, while all of the other lake whitefish populations and the North Sea houting, of course, are North Sea populations occurring on the west side of the Danish peninsula. This topology also showed a significant genetic contribution in the shape of migration from 2 Danish lake whitefish fjord populations into the North Sea houting population, sometime following initial divergence. Now, previous studies based on microsatellites and mitogenome data suggested that the evolutionary history that separates the houting from the lake whitefish is quite shallow. Estimated divergence time is less than 3,000 years ago. These results were based on, well, a simplified model of divergence with migration. But now, with genomic data, there is a possibility of testing much more complex scenarios, and therefore likely more realistic scenarios. So I tested multiple models using FastSimCo, and based on the best models, all of which included multiple size changes in the ancestral population, I found that the North Sea houting became its own genetic lineage already sometime around 7,500 years ago. Regardless of which lake whitefish population I was comparing to the houting, however, these models always suggested low levels of migration or gene flow between populations following this initial divergence event. And unfortunately, I wasn't able to obtain any reliable estimates of when exactly this gene flow ceased. Returning to this framework and the question of genetic exchangeability, based on the provided information, there's really no doubt that we should reject the hypothesis of contemporary genetic exchangeability. At present, Houting and lake whitefish are just not exchangeable. And I acknowledge that the symbols used in this framework are not very intuitive, so I just want to stress here that a plus sign means that we are rejecting the null hypothesis of exchangeability. The answer to the question of historical genetic exchangeability is not very clear-cut in this case. One could argue both for and against rejection of this hypothesis. So I'm actually just going to let this question stand for the moment. And then we're moving on to the question of ecological exchangeability. Testing this has really constituted a major challenge. Crandall et al., the authors of this, this framework, well, they recommended focusing on identifying ecologically important phenotypic traits that had a documented heritable basis. And that was undoubtedly the most realistic option at the time of writing this. It's quite an old framework at this point. But now with genomic approaches, we have way more accessible ways of testing ecological exchangeability. Genome scans permit identification of just individual genes under selection and provides direct insight into local adaptation and adaptive divergence between populations. So here you should really know that the North Sea Houting is the only corygonid fish in the world that tolerates high salinity oceanic seawater. So I have mentioned previously like anadromous fjord populations of lake whitefish, but these really only tolerate brackish water. And there is this recent study that did find evidence for physiological adaptations related to osmoregulation in the North Sea Houting relative to like the anadromous lake whitefish populations, but it was unclear whether there was a genetic basis for this adaptation. So that's what I set out to examine. This is just an example of one of the many outlier tests I performed comparing the Houting and the lake whitefish populations. And among the genes located in the outlier regions that I identified, there was significant enrichment for functions that related to ion transport and osmoregulation. So that implies directly that there is adaptation to a high salinity environment in the Houting. So it should be noted here that I did also find genes with similar functions under selection in some of the lake whitefish populations, but they weren't nearly as abundant. So that again fits the picture of the Houting going into oceanic seawater and staying there for years, while the lake whitefish, they just tolerate brackish water at best. Based on this information, again, there's really no doubt that we should reject also the hypothesis of contemporary ecological exchangeability. Given this unique local adaptation of the Houting, it is not ecologically exchangeable with lake whitefish at present. So the question that remains now is, when did this local adaptation arise? As you hopefully recall, I found that the North Sea Houting diverged from lake whitefish just around 7,500 years ago. If you happen to be well versed in Danish geological history, you know that this timing is uncanny. Namely, the Wadden Sea in itself, the home of the North Sea Houting, started to emerge just around 8,000 years ago. So this really suggests that the evolutionary history of the North Sea Houting is interlinked with the Wadden Sea, and that adaptation to seawater has been around for some time. So that led us to also reject this hypothesis of historical ecological exchangeability. And according to this framework by Crandall et al., this leads us to a recommendation of either treat as distinct species if we don't reject this question of historical genetic exchangeability, or treat as a long-separated species if you do reject it. So in conclusion, we really just determined that the North Sea Houting should be treated as a separate conservation unit, regardless of how you stand with this question of taxonomy. And this has some very important implications for like future conservation, practical conservation of the Houting. Because these same adaptations that make it a high priority in conservation can also pose some problems practically. Analysis of runs of homozygosity namely indicated that the Houting is moderately affected by inbreeding coefficients upwards of 10%. So it's not as critical as one might think based on the population decline. But some of this detected inbreeding has occurred within the last few generations. This is evident from some very long runs of homozygosity found in 2 individuals especially. And it's textbook knowledge by now that inbreeding can really only be undone through genetic rescue, introduction of unrelated individuals from other populations. But since there's only one population, one extant population of North Sea Houting in the world, well, genetic rescue would have to entail assisted migration from lake whitefish populations. But now that we know there is this adaptive divergence between the Houting and the lake whitefish involving salinity tolerance, this genetic rescue would impose a high risk of outbreeding depression. So this initiative wouldn't be recommended management-wise. So what's left to say is that by now it's particularly important to avoid further declines and bottlenecks in this extant population, because it's absolutely necessary that we avoid further accumulation of inbreeding and inbreeding depression in the Houting. With that, I would like to thank Independent Research Fund Denmark for funding my PhD studies, my co-authors, my supervisor, Michael Hansen, who could unfortunately not be here today. Thank you for listening. And if you are interested in knowing more about genetic rescue, please come see me at my poster